obviously there is a lot going on in regards of regulatory acceptance of microphysiological systems right now. So I thought it would be a good idea to talk about that a little bit today. Um, Let me see, I guess you can see that. So if you were online or maybe on LinkedIn in the past 4 weeks, you might have seen the announcement from the FDA about their roadmap to reducing animal testing and preclinical safety studies, with the first step being antibodies, basically, which I would say is not really the bread and butter of cardiac safety pharmacology, in fact, but actually it's just a first step and should be more modalities coming up in the near future. But at least it's a very soon step after the 2023 FDA Modernization Act, which was celebrated quite a lot. But so to, like, frankly speaking, I would say what they did like 2 years ago is just like add a little bit of new vocabulary to the regulation, which is like cell-based assays, microphysiological systems, bioprinting, and computational models. And well, if you do a quick search on the number of publications on these buzzwords from let's say the past 35 years or so, what's quite obvious is the fact that cell-based assays, the most important or the most basic of these terms, actually was on the falling edge again. No, I'm not seeing that, but you will be probably on the falling edge for like 10 years already until it made it into the regulatory guidance. And that may have like two reasons. The first reason could be that people are now using more specific vocabulary, more specific terms like microphysiological systems or bioprinting. But it also might be that actually it takes long, very long, the integration of these technologies, new technologies. And so it's even better to see now that it's increasing in pace. But still the question is, how can we as technology developers and users can accelerate the integration of new technologies in drug discovery? And we at In Vitro basically see two major paths for that. The one is to increase the physiological relevance of cell-based assays, so keeping on developing. And the second path is like demonstrating their fitness optimally in ring trials with regulatory agencies, academia, and industry. And I would now like to address both these paths that we're going down the road with all of you guys. So when we talk about increasing physiological relevance, especially in the field of cardiovascular sciences and safety, the biggest buzzword is maturation. So maturation is the topic with human iPSC-derived cardiomyocytes. And so in the past couple of years, a lot of technologies and ways to improve the maturation were developed and established and made it actually into the scientific community. I would like to address some of them, starting with substrate stiffness, so giving the cells a flexible substrate to contract, really. Another parameter is prolonged culture time. And if you combine that with others, you might even decrease the prolonging that you have to do, because if you have to culture the cells for several weeks, many months, that's not really ideal. But combining some of the other technologies can help that you do not have to prolong it for that long time. Another approach to increase maturity is biophysical stimulation, either mechanically or electrically. And beyond that, cardiomyocytes under physiological conditions are never alone. They are always surrounded by fibroblasts and fibrocytes. So co-culturing them might also help increase the physiological relevance. And finally, if you add fibroblasts and other cells, then you are already close to running these assays in 3D cultures with organoids, with spheroids. So let's start with the substrate stiffness and some ideas, and that also relates to 3D organoids and cell-based assays that were already used before. So imagine you have your cells seated, not really seated on a substrate, but in suspension culture, just like an idea, like a theory. The only thing that this that cardiomyocytes can do is contract once, because they do not have any apparatus that can relax them actively. So they can just contract once, and that was it. That's what we call isotonic contraction. The other extreme is actually what we see with regular cell culture plates with very high stiffness. Or like infinite stiffness. In this case, the cells go from zero stress to maximum stress with each cycle of contraction and relaxation. So they are not within the physiological range. And so the physiological relationship is actually when they have like an extracellular matrix or something that keeps them in the physiological range of stretch, and that is essentially when you seed them on flexible substrates or keep them in 3D cultures. So all these anchors and gauges and springs that you see here actually have a real representation, a biological representation. And that is what they use when they sense an environment like you see in the video on the left side. That's a 3D culture on our membranes. And it really shows you, it gives an indication what the cells really see and what they experience. And they experience this soft substrate through three major routes, basically. The first one is integrins, so the adaptive proteins from the inside of the cell to the outside. The second one is receptor tyrosine kinases, and the third one is ion channels. And each of them have certain subgroups that are stretch and stress sensitive. And if they are outside of their physiological range, they have a negative impact, like on gene expression and on metabolism, and by that changing everything that's happening in the cell. But it does not just remain on the inside of the cell, because cardiomyocytes also interact with their surrounding cells, like fibroblasts and fibrocytes. So they can activate fibrocytes to become fibroblasts and remodel the extracellular matrix. And once the fibroblasts remodel the extracellular matrix, they change the mechanical environment of the cells. And by that, like a positive feedback loop, have an impact on what the cells sense through integrins, receptor tyrosine kinases, and ion channels. So this is a big feedback loop, and that actually needs to be well determined and well controlled if we want to replicate what's happening in the heart in vitro. So let's take a short look. I will try to keep it short here, because it's not just that we're already behind the time, but I also need to get the train to Paris to a conference. I will try to keep it now as short as possible, maybe run through one or two of the next slides. So let's look at the integrins and the other adaptor proteins. So actually, cardiomyocytes can sense fast muscle myosin contraction and slow non-muscle myosin contraction through integrins and adaptor proteins like talin. And these proteins like talin has a threshold of 5 piconewton. If it is stretched below that, or if it's stretched above that, so it changes its conformity. And only if it changes this conformity in a very regular way, like the cells beat, like every second or something, then there is positive pathway activation downwards, or at least no negative pathway activation downwards to other protein kinases that are inside of the cell and not kinase receptors. Another interesting finding from Heras Bautista and others at the University of Cologne a while ago um, was that actually cardiomyocytes respond with massive deregulation of metabolic processes when they are cultured on wrong substrates. So what they did is they seeded the cells on polystyrene, so usual cell culture plastic, on very stiff gels with 123 kilopascal that resemble fibrotic tissue, medium 30 kilopascal gels that resemble usual cardiac heart tissue, and very soft gels. And they saw, of course, what everybody would expect. The stiffer the substrate, the more deregulation you have. But interestingly, it's basically and predominantly cellular metabolic processes that are changed by that. And last but not least, ion channels. Like, we are all so much into ion channels and dealing with them all day. I guess, like, I'm sure everybody in this meeting here. But what we are not so aware of, what we should keep in mind, is that ion channels, even L-type calcium channels, are stretch sensitive. And Takahashi and colleagues made some nice experiments in H9C2 cells. They stretched the cells and they saw that there was calcium influx into the cell. And so they hypothesized that this might be due, at least partially due, to stretch sensitivity of L-type calcium channels. And they tested that with L-type calcium channel antagonists like nifedipine, diltiazem, and verapamil, and they saw, yes, you can reduce this stretch-induced peak in calcium by application of these antagonists. And what they also did is use siRNA to knock down L-type calcium channel expression. And again, they saw that the calcium influx was reduced. So, and that's very, very good proof that even L-type calcium channels are stretch sensitive and need a proper environment, physiological environment. Okay, so far regarding the first thing, the first path, which is improving physiological relevance. So we know we have to improve physiological relevance to get proper maturity state of the cells. Now let's come to the second path, which is the ring trials and the demonstration of the technology to the academic field and also the regulatory field, of course. And so the first big study that we took part in was the HESI cardiotox study recently, which was the successor study of the famous CPER study. And in this study, there was a little bit different approach, which I will discuss in a second. So what we did in this study, we used the FlexSite technology that we co-developed with Nanion Technologies to measure contractility. So what we do there basically is we seed human iPSC-derived cardiomyocytes on ultra-thin silicone substrates that mimic the mechanical environment of real human heart tissue with an elastic modulus of approximately 33 kilopascal. Now, the membranes bend downwards by the weight of the medium, And once the cells attached to the membrane start beating synchronously, they lift the membrane upwards. And as soon as they relax, the membrane goes back down. And this movement is measured by means of capacitive distance sensing. So beneath each membrane, there is one electrode that measures the distance or the movement of the membranes that you can see on the left side there. And from that, we derive a set of parameters, including, of course, contraction force, which is the twitch amplitude here. We measure, of course, beat rate and beat rate regularity, as well as the occurrence of early after contractions. We can take a very close look into the beat duration on certain levels of the contraction relaxation cycle, or actually on every level, whatever is necessary. We look into the slopes of contraction and relaxation. That's especially interesting if you want to get an idea if a compound has effect on certain ion channels if you do not have the capability to patch that. And also, like, we can look into secondary parameters like contraction and relaxation area under curve, but this is not what we're going to discuss today. Another technology that we used in this context of the HASI cardiotox study was impedance spectroscopy with the CardioExcite 96 by Nanion. So in this case, the cells are seated on gold electrodes on stiff substrates, but still we have very many advantages regarding the parameters that we can measure, because we use this technology here to measure the monolayer integrity. So once the cells, for those who are not familiar with impedance spectroscopy, once you see the cells on the gold electrodes and the cells spread out, they will increase the base impedance until it reaches a certain plateau. And if you reach that plateau and then add a compound that's, for instance, cardiotoxic, and the cells die off, they shrink and get apoptotic, the base impedance goes back down. And this is a nice measure for monolayer integrity. And these two technologies were applied by us in this study. And the basic idea, the basic question of the study was, can human in vitro models predict subchronic cardiotoxicity? While the CEPA study was very much into very acute effects, here in this case, the question was, is it possible to do it subchronically? So a decent number of test sites collected for this ring trial. In total 23 test sites from all industries, pharma, biotech, CRO, and academia. A total of 10 assay platforms was used, ranging from electrophysiology through mechanobiology, optical methods, and also biochemical endpoint assays. And as I said, we applied two of them, impedance spectroscopy and contraction force analysis. And a total of 12 blinded compounds was tested. So they were sent out to each site blinded. And these ones, the compounds targeted like energetics or mitochondrial toxicity, electrophysiological disturbance, contractility, and structural and myofilament disturbance. Let me take a look. 5 off. So maybe I'm not going to show all the data to you so that we can keep the time a little bit. That's the experimental outline that was used, just very briefly. So the cells were pre-cultured for 6 days. Then there was a media change to serum-free conditions to account for serum binding and other effects of serum in assays, which is also quite debatable how good serum is. However, so there was a change to medium to serum-free. And then 24 hours later, first compound application, and then the first 1 hour measurement. So 1 hour to account for acute effects, to see if there are acute effects or not. And then a total of 5 days in our case. So we measured the cells for 5 days. The most important time point, and which I am referring to now on the other slides, is the 72-hour time point, because there was also endpoint assays, biochemical endpoint assays, and there you could not do like repeat measurements. So that time point of 72 hours was the one that was most important, basically. And that was then used to evaluate the effects of the compounds. Maybe very shortly, so I really like to see all those different time effects. So like vinorelbin obviously had only a dose-dependent effect, no time-dependent effect, or at least not at the highest concentration. So there, after 1 hour, the effect was already there. With the lower concentrations, it took a little bit longer, like let's say 24 hours until the total effect was there. In contrast to that, doxorubicin, a very well-known cardiotoxic compound, had both time and dose-dependent effects. So the higher the dose, the steeper the curve here. So the quicker the effect, lowest concentration took a little bit longer. And like BMS 98, 60, 94, really interesting. Never seen a kinetic like that. I have no idea why it looks like that. So it's a mitochondrial toxic compound, but why it looks like that, you have no idea. Super interesting for me. Another look on the watch. Okay, so I'm not going to bother you with all these data. Maybe just run through it a little bit. Doxorubicin, super cardiotoxic, highest two concentration, killed everything. Erlotinib, known as a very cardiac safe compound, showed this safeness, this safety here, while sunitinib, known as very cardiotoxic, also showed this cardiotoxicity, also both on the functional as well as on the structural level, which you can see up there. Pentamidin, also interesting because it's a time-dependent effect, so it doesn't affect instantly. Showed exactly that effect both here on the functional as well as on the structural level. And yeah, well, let's skip the next one because I need to get my train. Let's conclude. Subchronic assessment of compound reactions is possible with human iPSC-derived cardiomyocytes. Compound reaction kinetics can be resolved with cell-based assays in subchronic timescales, and we can distinguish between functional and structural cardiotoxicity with different cell-based assays, but all in a 96-well format. Now, briefly, another really nice study that we were part of was the Botanical Safety Consortium study initiated by FDA in Nice. I really love to see these botanicals. So that was actually the compounds that we tested. There was botanical extracts, so many nice colors. I really love to work with that, and also all these interesting extracts, like from aconite, ephedra, everybody could know, oleander, thundergold vine, yohimbe. However, very nice compounds tested here. This study was rather small, only 6 test sites, but also from all different parts of the industry. And we did contraction force, and here you can see really nice that we basically could really predict the toxicity, because aconite, oleander, golden seal, yohimbe, that were all positive controls, while comfrey, kratom, ashwagandha, and thundergold vine were more like the negative controls and had little or no effect. Interestingly, I'm going to show just this one here. Green tea, we used it very close to the solubility level, solubility limit, basically, because we wanted to see in this study, do we actually see anything at all? So the highest concentration under acute treatment of green tea, decaffeinated green tea, had an effect. But none of the other concentrations. Interestingly, after 24 hours, even green tea had concentration-dependent effects on each parameter that we analyzed, but we have to keep in mind you can never drink so much decaffeinated tea to get blood concentrations like that. So, but anyway, interesting finding because like ashwagandha had absolutely no effect here. However, let's take a short outlook. We now just looked on pure cardiomyocytes, and as I said before, the interaction with fibroblasts could also be interesting. We have a very nice collaboration with Fujifilm CDI, where we tested their fibroblasts and cardiomyocytes in co-cultures. What you can see here is there is a dramatic increase in contraction amplitude if you just add 10% fibroblasts. So they have very, very beneficial effect on the cardiomyocytes. And that beneficial effect also remains over the whole period of time. And now if you like combine cardiomyocytes and fibroblasts, then of course the next idea is bring them into 3D. So this is the results from a nice collaboration with University of Cologne, Marga and Walter Boll laboratories. So we see that like single organoids on the membranes can measure really nice singles, although there's just tiny little organoid on this rather huge membrane. It's a 96 well membrane, where it's really large compared to the diameter of these organoids, but still we can get really, really nice results. And if you like seed many of them and let them come together, they will form something like you can see here. That was all of that was like tiny little organoids, but they fuse together, and you can really go 3D here. So that's outlook, what's going on right now. And if I remember right, that's it from my side. I don't know if we want to take questions, Elena.
Yes, maybe if you have time, maybe we can have one, maybe or two.
Yes,
If anyone, I don't.
I can
Yes.
run to the I can run. We can do one. Yes.
Okay, one. Okay, then I'll be good to ask first and very fast.
Right.
You showed a very nice study concerning the mechanosensitive ion channels. Did you ever collect cells after them being on the flexible membranes? And did you look at, I mean, apart from calcium, maybe piezo channels or any other mechanosensitive channels?
Piezo actually not yet. So I know that there was gene expression analysis done in the past, but I think they were not intended to be published. I don't know, because there's always this legal stuff with human iPSC-derived cardiomyocytes and ethical stuff and everything. So it's always hard to then publish this data. But it was, yes. And
Mm-hmm.
really interesting findings about the regulation of, especially of like calcium channels and sodium channels, and downregulation of funny current